Everyone will end up in a...
Yeah. Hi, Stefan.
Okay, hello everyone, and good morning to the second version of this ACCS OSPM webinar and workshop series on data collections today. And before we get started, I would like to acknowledge the country on which we're on, which are the Turrbal and the Jagera people for us here in St. Lucia. And the plan for today is we have a couple of local speakers in Australia. So today we don't have any international speakers, and they will show how they work with large open access data sets on CVL, on M3, and then also other users of collection data. So we start with Farnoosh, who will give an introduction to data collections in this ACCS landscape. So what this project wants to do with data collections and where this project wants to bring these. Then we have Stuart, Oldham, physiological constraint generative models of structural brain networks, then Kevin on denoising, Jurjin on characterizing scale-specific cortical shape symmetries. And then we move on to other collections, Data with Eli Müller and Aaron Warren. And then we have a quick break, because that's probably a lot of input. And then we move on to an hands-on component where Catherine and Chow demonstrate how to use some of these data sets on CVL. So I think that will be quite interesting today. Then if you think this is really interesting and you want to do it yourself, then there will be a real hands-on workshop, which will be on Wednesday, the 15th of December, 2 to 4 p.m. And this has limited spots because this will be really interactive and you will be really working on CVL, how to access. First, you get to know what data collections are actually there, how these data collections can be accessed, and what you can do with these collections. And if you haven't signed up, I think there's still spots left, Catherine, right? So she will post that link in the chat now, and then you can sign up. Then the last thing from my side is we are already planning the next webinar, which will be in February 2022. And the topic will be going beyond group average single subject analysis, which was the winner of the survey we did a couple of weeks ago. So that got a lot of votes, and we said, okay, let's do this next. We don't have confirmed speakers yet, but we have exciting ideas there. So stay tuned and we'll announce it early next year once we contacted speakers. With this, I hand over to Farnoosh.
Thank you, Stefan. Can you please share my screen? Thank you. Can you see my screen now? Great. Hi, everyone. My name is Farnoosh Salapian. I'm at Monash University. I'm data engineer working at Monash eResearch Centre and Helix platform. I'm part of the ACCS project, the Australian Characterization Common at Scale. And today I'm going to provide you an introduction to data collections in the ACCS landscape. We've provided a couple of webinars and presentations at OHBM regarding the Australian Characterization Common at Scale project. So I briefly touch base on this. It's a national scale project to accelerate image processing analysis and publication. It's funded by ARDC and following partners, as you can see in this slide. And it has a few, it has three specialized work packages, and one of the work packages is work package 5, which is about biomedical imaging collections and analysis. In this work package, we have three streams of work. We have the one stream of work which is about training, which is such as today's webinar is a collaboration with other organizations such as OHBM, ISMRM, and other organizations where possible to provide training to neuroimaging community. And the second stream of work is to provide access to biomedical imaging data collections and the capabilities to provide them, to process them. And the third stream of the work for us is to provide infrastructure to multi-node Australian data collections across Australia. The third stream is collaboration we have where possible to with other projects, like, for example, Australian Imaging Service or BioCommons. Today's presentation is focused on the biomedical imaging data collections, and how we manage the data and how we offer the commonly used data collections. What are the data collections that are available on CBL sites, the characterization referral laboratory sites at Massive, for you to have access and use it for your research and process your data. What's the benefit for researchers? You can access to terabytes of commonly used data collections that can be used centrally, that are managed centrally for for authorized users to use it, and it's located on the servers on HPCs, high performance computing systems, for you to process data. For more computationally intensive image processing pipelines. We take care of the data management side of the work. We go through the planning to storage. We make sure that there are enough description about the data collections that we make available to researchers. Ensure that if the backup is required, if we need to update the data, if the version control needs to happen. And if also we make sure that the data is anonymized and is not identifiable. And when it gets to the usage and publication, we need to make sure that we are following the data use agreement that's defined by the data custodians of these data collections. And technically, we can support those data access and provide access to our researchers based on those limitations. And when it gets to the retention, and for example, if it's time to decommission a data collection, we make sure that the users are in the loop. So we don't go and delete the data. We make sure that the users of those data collections have been contacted before we decommission the data, for example. So we go through all of this process for researchers to be able to access this data and do the processing and publish the results. Here are the list of data collections that we manage. So the left side is the curated data collections, and the right side are those data collections that we provide infrastructure support to. As I said, the left side is more of a focus in this webinar. So I go quickly one-on-one through the data collections that we have available. These collections are currently available at Massive, but all Australian researchers can access to these data collections, basically, as long as they go through the access procedure. The first data collection, which is a very popular one, is the Human Connectome Project dataset. We have over 50 users at the moment using this data collection at Massive. It has a quite comprehensive data. It has a 3T MRI imaging of over 1,200 healthy young adult participants. Most of them actually have structural MRI data. A subset of them, about 180, has 7T MRI data. About 90 of these subjects have resting state MEG and task MEG data collections. And again, subset of these subjects have repeated 3T MRI data and behavioral retest data. There is a specific access procedure that my colleague Chao, who's working with me on the work package 5, will go through it at the end of this webinar. And you will see a few of the presentations in this webinar how researchers use HCP data, either on Massive, CBL, or other HPCs across Australia. We also host a couple of other data collections. It's called a Human Connectome Project Lifespan, Aging and Development data collections. Each of these aging and development, each of the data collections has about 700 data with different age range of subjects. Those are all healthy participants. We host subset of this data because the structural pre-processed data as well as resting state fMRI pre-processed and behavioral data. it's in general, there are like about 12 terabytes of data that are available for you to use. It has a very particular data access procedure. It's very restricted, although it's considered as open access and reference data collection, but it is only provided access to eligible researchers. So it is provided access to individuals or group of researchers. You have to go and agree with the terms and conditions, sign the data use, have the data use certificate, and when you have all of that in place, you can contact us and we will add you to the appropriate user group that can access these data on our website, on our HPCs. Another data collection is the Human Connectome project for early psychosis. It has unprocessed data of all modalities, structural MRI, resting state fMRI, and diffusion MRI of 183 subjects. Those subjects are with age range of 16 to 35 with early phase affective or non-affective psychosis with an early onset of psychotic symptoms. There are also minimally preprocessed structural MRI available on most of these subjects, and also clinical and behavioral data of these subjects are available. The good news is that if you have access to the lifespan data collections, you can use this data as well. It's again above 2 terabytes of data. The next data collection is Nathan Kline Institute Rockland sample, the neuroimaging release. It has MRI data, physiological data obtained during scanning, and only basic phenotypic data of the subjects. The information about data access procedure is available on our website in 3 documents, and the link to the original website is also available there. It's open access. It doesn't have any licensing agreement in play requirement, and as long as you send the access request through our Kuragi system on N3, you will be added to the user group and you can access to this data collection as well. The last one is the Brain Genomics Superstruct project, which is a collection of neuroimaging, behavior, cognitive, and personality data for over 1,500 human participants. It includes high-res MRI data acquisition and resting state fMRI data. And each functional acquisition has gone through the QC pipelines, and pre-computed brain morphometrics are also provided. We have the latest version of this data collection, and again, the data access procedure for this data collection is available on our website. You have to agree to their terms and conditions, send us the access approval that's given to you by data custodian, and we will add you to the appropriate user group to access this data collection on CBL sites. And at the end, I'd like to invite you to use these resources. For more information about the open access data collections, the Nathan Kline GSP and HCP data, you can get more information on the N3 website here. And for getting information about how to get access to the more restricted data collections, such as the lifespan data collection, we're very happy to provide you the information. We already worked with one of the research groups on the steps that they need to take to apply for the access. We're very happy to help you as well on that. Please contact us if needed. And there are other links to CBL and also Imaging Tools website, which is the newly created ACCS related. You can find the ACCS related activities in that website. And if you're interested for us to host or manage other data collections for you, please reach out. We will do some analysis, and based on that, we will work with you and make that available possible. Yeah, thank you to all the ACCS project team and also the HPC team at Monash who helped me a lot in making these data collections available. Thank you so much, Catherine, for sending the links in the chat. Back to you, Stefan.
Yeah, thank you very much. And over to our first speaker.
Great, thanks, Stefan. Let me just share my screen. Great, you can all see that?
Yes, perfect. Thank you.
Awesome, right. So thank you very much for the opportunity to present today. So today I'll be presenting some of my PhD work, which is about the modeling of human brain networks. So a brain network is a representation of the brain's neuronal connectivity. There are numerous ways to construct a human brain network, and I'll primarily be discussing it in the context of a structural network. So in a structural network, what we do is we use diffusion-weighted imaging and run a process known as tractography to reconstruct potential white matter pathways. We can also parcellate the brain up into different regions, and we can combine these two melodies to make a network by having each region form a node in the network, and then we assess if streamlines connect the region to a pair of regions in the network, and if so, we form an edge or a connection between the respective nodes. And then when we have this network representation, we can begin to ask questions about the organization of the brain's connectivity or its topology, the arrangement of connections. And so we can identify things like modules, clusters of nodes which are densely interconnected between themselves, but weakly interconnected outside that cluster. And also things like hubs, where hubs are highly connected nodes. So hubs have high degree, high number of connections, and are important to brain function. And in network neuroscience, we're very good at describing all these various kinds of topological properties and how these relate to brain function. But increasingly, we begin to move towards asking, how does the brain actually get these particular kinds of topological features? What kind of developmental mechanisms or constraints are there on how connectivity forms? And this question was originally considered by Santiago y Cajal, arguably at the dawn of modern neuroscience. And he proposed these 3 laws of conservation governing how neuronal connectivity forms. And these laws are of time, space, and material. The conservation of time means the nervous system develops to minimize neuronal conduction delay, meaning a nervous system needs to promote efficient communication or have some kind of functional efficiency. The latter 2, space and material, indicate the need to minimize metabolic demands. So you can think of each neuron as an energy demand that's proportional to its size. This suggests that there is some kind of wiring cost to forming a connection, and that cost is dictated by the size or length of the neuron. So on this latter point of wiring cost, how can we see how to the extent to which this shapes brain connectivity? Well, one way is we can use generative models. A generative model is pretty simple. It just says that the probability of connection forming between 2 nodes, in this case Pij, depends on some exponential function of the distance between them, Dij. And this function is controlled by this eta parameter. So under this particular configuration of the model, we will get mostly short-range connections, but some long-range connections. And then we can adjust this eta parameter to change the extent, the strength of this distance penalty, such that we only get mostly short-range connections here, or we can relax it and get more and more long-range connections. And so this is one way which we can model this, but you'll remember from my earlier example that in addition to wiring cost, there's also this idea of functional efficiency. So how can we incorporate this into this simple generative model? Well, we can add an additional term to cover functional efficiency, and this is usually initialized as a term favoring a particular kind of topological arrangement. And we refer to this as a trade-off model because we have a wiring cost competing with functional efficiency, and both of these 2 factors drive the resulting connectivity. And so what does a kind of topological arrangement look like? Well, for instance, one prominent one is this idea of matching, the matching index, where basically if 2 nodes have similar neighborhoods, they're more likely to form a connection. And this trades off with the wiring cost to form connections probabilistically. And there are many possible topological rules you could have. Here are just some of them, and we'll go into this a little bit more later on. So you've made this network using this generative model. How do you know how well it's replicated the underlying brain data? So what we do is we compute a number of topological properties. In this case, we look at the degree distribution, some of connections each node has, and the edge length distribution. And for our data in the model, we compare the empirical distribution functions. And we do this across 4 different topological properties, which are considered somewhat characteristic of brain networks. and we look at the maximum discrepancy between these curves across these 4 properties, and that becomes our indication the model fit. So the smaller this discrepancy is, the closer our fit. And then we use some sort of optimization scheme to find the best parameters that produce the best fit. And so current results sort of show that these homophilic mechanisms that were similar to that topology mechanism I was showing you earlier give you the best model fits. And this geometric model, which is only using wiring cost, is giving the worst performance. And this is sort of where the field is at currently. But we thought there's some issues with generative models which we wanted to explore, of which I'll be highlighting 2 today. So the first concerns biological plausibility. So these topological measures, which we're including in the term, they're somewhat abstract, and it's hard to link to underlying physiological mechanisms. So we wanted to see if we could include more biologically grounded constraints into the model. And the first thing we thought of was geometric changes. So when your brain is forming connections, which largely happens in the second half of gestation, the brain's a lot smaller. In fact, 21 weeks is about 20 times smaller than the adult. This means that distances are a lot shorter, and so this might mean that a long-range connection is more likely. So there's going to be an altered wiring cost. And in particular, across development, the brain increases in size. And so this increase in size will again change that wiring cost. So we wanted to see if incorporating this into our model could alter its performance. So from MRI scans of fetuses in the womb, we extracted on a week-by-week basis from 21 weeks to 38 weeks a surface representation of their brain. And basically, for our model to work, we need to know where the position of nodes are across all these times so we can measure the changes in distance between them. And so on our adult, we have our parcellation for our adult data. Essentially, we need to project it onto each baby brain. And the way we do this is the procedure known as multimodal surface matching, where we do a spherical projection of the surfaces to a sphere and then align them based on cortical features. And when you do this alignment, you can project one brain into the space of the other. So we can take our adult brain and project it into the space of any of the baby brains. And this is handy because, as I was showing you before, on our adult brain, we have our parcellation defined. And so because we basically know through multimodal surface matching where each point lies on each of these baby brains, we can then project our parcellation back through time and get indication of where each region is. And so we can measure the distances between all those regions at every time point, and we can incorporate into our model pretty simply. So we have a growth model where we use the distances at one time point, form connections, move to the next time point, form connections, and so on and so forth. Then we have a static model, which is just purely using adult distances. And this static model is what most current studies in the literature use in their generative models. The second thing we thought for a biological constraint was, in the literature, there's been a lot of work showing that the psychoarchitectural similarity of 2 regions is associated with an increased connection probability. And this relates to the idea that if two regions are developing at a similar period, they're more likely to have similar psychoarchitecture and are also more likely to form connections with each other during this period. So we can use similarity as another potential constraint in our model. And so we do this in a number of ways. So the first way is we use correlated gene expression, which is just the similarity of each region's gene expression. And we use it in uncorrected and corrected form. So there is a spatial order correlation present in this data, that's in the uncorrected form. And in the corrected form, we remove the spatial trend. We also use more direct measures of cytoarchitecture. So we use from histology data, indicating the pairwise similarity between each region. And also we use something called the T1, T2 ratio, which you can consider an index of myeloarchitecture. And so we can include these similarity measures simply into a model like this. So for example, for corrected correlated gene expression, we can just make the probability dependent on the similarity between each region. And we can also combine this with our wiring cost term and have the spatial constraint as well. Finally, the next thing we wanted to sort of investigate was this idea of spatial properties. So I've currently described that the way we sort of test our data is we look at how well it replicates these statistical distributions of topological features. But the brain is more than these distributions. topology has a distinct spatial embedding. For instance, if we look at the hubs, we know the hubs are located in very distinct places, and we know that their placement is very important. The brain would have very different dynamics if they were located in different positions. So even though our model might be very similar on these statistical distributions, we can see that it can have potentially a very different spatial distribution of topological properties like hubs. And this would be a big problem because these spatial properties are very characteristic of the brain. So we wanted to see whether generative models could capture the spatial embedding of topology. So to do this study, we used those models I've been describing, and we performed it in 100 structural connectivity networks for the left hemisphere constructed from the Human Connectome project. So this is a data set of a full 1,000 people, which we processed on Massive and have openly available. I only used 100 simply because all the results I'm going to show you took about a month to run, and I wanted my PhD to get done sometime this century. So in the interest of that, I focused on a small subset. And first up, I'll present the results for looking at our growth models and our physically constrained models. So the way to interpret this figure is on the x-axis, we have different models, and on the y-axis, we have model fit. And remember that a smaller model fit member indicates a better replication of the brain network. And so to break this down, first off, we have our spatial model, which is again using only that wiring cost model. And we see that growth gives a very small, and I mean very small, improvement in model fits. And if we look at a topological model, in this case, the matching index, we see that there is more of an improvement when using the growth. So it seems that the topology is able to take advantage of these smaller differences, of these altered length scales. But you'll see that, of course, the difference is quite small. And the reason we discovered for this is the model is actually compensating for the smaller distances. It's imposing a stronger distance penalty. And this distance penalty is largely be able to counteract a lot of the changes in distance. So next, moving on to the more explicit psychoarchitectural similarity models. So we look at things like our histology and T1 and T2 ratio, and we see that some of these are performing similar to the topological model or even slightly outperforming it. And this is good because these models are more biologically plausible. It's more easy to relate psychoarchitectural similarity to underlying biology than it is topology. And it seems as though the T1, T2 ratio gives slightly better fits than the histology data. But the interesting one for us was gene expression. So if you use it in its corrected form, so corrected for a spatial autocorrelation, correlated gene expression by itself gives the worst performance. When we combine it with a wiring cost term, it improves a lot, almost doing on par with our matching topological models. But when we use it in its uncorrected form, so this has an inherent spatial autocorrelation present in the data, these models are giving the best fits. And of interest is this single uncorrected correlated gene expression model, because this is only a one parameter model. All the others are, all these ones, for instance, are 3 parameters. And so this very, very simple model is giving us very close to the best performance. And we think that's sort of interesting and something to explore more in future. And next is to think about is the spatial embedding. So first we want to look at how well does topological models, the best topological model, replicate the spatial embedding. So here I'm showing the similarities in their empirical distribution functions, and you can see they're very, very close. And here's what the empirical distribution of degree looks like. And as for our model, it's not close at all. There is a mismatch. In fact, if we look across any of the 4 properties that used to make up our model fit function, that the model isn't replicating the spatial embedding of any of these topological features. But what about our best physiologically constrained model? Will that do any better? And the answer is still no. Some things are moving more in the right direction, but it seems like no generative model, no matter how you format it, is able to capture the spatial embedding of topology. So there are subtleties here to unpack. So firstly is that growth does give some small improvement over static models. Again, emphasis on the small, and largely this is just because, as I said before, the model is compensating for these smaller distances. Physiologically constrained general models show promise, particularly correlated gene expression. And again, just to reiterate, this is likely related to the idea that this is reflecting similarities in developmental time periods. And this suggests if we were to more explicitly model this idea of heterochronicity, namely that regions develop at different times and different rates, then potentially that would allow us to more accurately capture the brain's connectivity. And finally, all generative models, no matter what, fail to reproduce the spatial embedding. Again, spatial embedding of topology is a really key characteristic of brain networks, and the fact that these models can't replicate this property is something that sort of needs to be addressed in future work. And so with that, I would like to finish up. I'd like to thank my supervisors, Alex Finito, Ben Fulcher, and Kevin Aquino for helping really shape and direct this project. Arina Arnakevich-Ute, Rosa Shishigira, and Casey Picola for helping me get all the data ready for this project. And finally to Massive for helping me to process and store all the data that this project needed. And thank you all for coming along today and listening.
Well, thank you very much for this great presentation. Yeah, with this, we move over to Kevin.
Cool, thank you. I'll just share my screen here. All right, I'm sure it's visible for everyone. Yes. Great. Okay, cool. So I'll be talking about denoising resting state fMRI. This is the work I did as a research fellow at Monash University, but I've moved on to University of Sydney and BrainKey at the moment. So basically, let's have a recap of what is resting state fMRI. So for those that don't know, resting state fMRI is an interesting functional paradigm where subjects are told to go to the scanner, lie down, and just let their mind wander. And this is really quite an interesting paradigm because it doesn't require much instruction, but even within that, if you look at correlations between parts of the brain, you find really nice distributed networks. And so here I'm showing one of those networks, which is called default mode network. And specifically here, if you take one part of the brain, the posterior residuum cortex, and look at the correlations between all the brain, you find this thing called the default mode network. And this is one of the networks you find when you analyze resting state fMRI. And this is quite the target of many studies because it's easy. It can be deployed to multiple subjects. So for example, the HCP has 400 of these. UK Biobank has 40,000 of these people doing these things. And also it can also be quite simple instructions for case control studies. And many researchers have found that there are differences in the spatial distribution and the different correlations between different case control groups. So it's a really a nice target for people to be looking at, I guess, human function. However, with all that nice features I just talked to you about, there's one big problem with resting state fMRI. It is horrendously noisy. It's full of artifacts. So it's a target of a lot of pre-processing pipelines. For example, there's people do very extensive pipelines to try to make this data as clean as possible. And when I say clean, I mean removal of noise that are kind of non-interesting. So for example, one thing people often see is the correlation of all these patterns to things such as movement or breathing, respiration, or heart rate sort of effects. And so to analyze this data, you get to denoise your data. But of course, to denoise your data, you check whether it works or not. And so what researchers have found is that a single QC measure isn't enough. You can't just apply some off-the-shelf thing and just have a summary effect of all your data sets. What you actually need is a way to visualize all these individually. And one of these things that are done, which has really changed a lot how we look at resting state fMRI, is visualizing what's known as a Kappa plot or graph plot of your time series, popularized by Jonathan Power in 2012. And so what that is that the x-axis, you can see here, is time, and the y-axis is voxels. And what is shown in these things is that even if you use the most sophisticated pre-processing pipeline with the most sophisticated imaging modality, you do see residual effect of these very large whopping, you can see here, these big bands of activity. And they all seem to be time locked with these motion measures, which is FD, which is frame-wise displacement. And he found that the only way to really look at this and to get rid of these big bands is to do something called global signal regression. And so essentially I take the mean across all these voxels, and so the expression at each voxel, and I remove that to form a residual signal. And and see the quite visually striking results of this pre-processing pipeline. However, we want to question that to see whether or not there's some artifacts, some extra things people are missing. And so I think I want to just bring on this point. My talk isn't as exciting as Stuart with animations, but my talk is really about looking at QC. This is my sort of this talk here. And so we really want to be investigating this properly. And so to do this, we of course use everything on Massive. And I guess in terms of the Open Science Framework, we download all this data from the UCA Phenomics cohort. This is available on OpenEuro. It now sits on Massive. We also use fMRI Prep, which is a nice container which contains a whole bunch of pre-processing pipelines all in one little container that can be shipped around to multiple nodes across Massive, to multiple computers, to make it very reproducible. And that's available on Massive, module load, fMRI Prep, and choose your version, that kind of thing. So it all sits there. And so to deploy this QC measure, we did the following things. We first looked at things such as frame-wise displacement you can get from your pre-processing pipelines to determine how much the subject moves. DVARS, another type of metric to look at your variance over time for across all voxels. And then we have to look at this color plot itself. Now, interestingly, to do this, you take a 4D matrix, which is what Nifty reads in from your functional MRI data. You've got to squeeze it down to 2 dimensions. So the way you do that, you can use something in MATLAB called Reshape. We go from 4D to 2D, or in if you're a Python person, you use Reshape, possibly in a NumPy. And so the way this gets unfolded from 4D to 2D, it's like a technical point, is actually not standard. Every other package has its own thing. So I'm going to call this a random ordering of how voxels are associated. And now you can also order these voxels by looking at some group average or order them by the community participation, that kind of thing. However, this is all based on group averages. You want to look at this at individual subject level. Reminder that the goal is to look at the QC across multiple subjects quickly. So you want to do this in the best way possible. And so instead, we want to relook at this to focus on individual subjects, grouping voxels by their signal similarity. So you can do it 2 ways. We're going to present 2 ways. Is to do this by reordering its correlation to the global signal. So i.e. taking the average of across all voxels and look at the correlation to that mean fluctuation. Or I could do some hierarchical clustering, which I'll call the cluster ordering, so CO. So GSO is the global signal ordering. So here's an example where I've got one subject. I've gone through my QC. I've generated this plot. I've got the movement, which is FD, the gray matter signal, GM, which is also the global signal. And what I'm going to do now, this particular plot, I'm now going to reorder it to make sure I emphasize signal similarity. So I'm going to order it by its correlation to the global signal or correlation to a GM. And as I press play, as I go on, all of a sudden now this gets rearranged and I can really emphasize all these artifacts that are potentially there. And essentially these 2 regions here, they're kind of time locked to various events of either respiration or here movement. So these are kind of, I guess, noisy structures you see there. I mean, there are 2 examples, but of course there are multiple ones throughout the whole session. But interestingly, they also occur in phases, like some of them are positively grouped together and some are negatively grouped together. If I then order them by hierarchical clustering, so CO, you can see now there are 2 big whopping bands that seem to be out of phase with another. So there are 2 different versions of looking at the data, or so 3, I should say, and each of them can show some, a new artifact you've not considered before. So by looking at this, you can then take all your data that we found in this UCLA cohort. You can categorize the data into 3 kind of subsets. The first one is a bunch of data which has what's called WFCs. I call them widespread inflections, so kind of globally correlated activity. Other data sets which seem to have very strong single phase presentation of these artifacts, or I call category 3, which has 2 phases of these responses. And that you can see that mainly with the global signal ordering. If I were to look at the cluster ordered plots, it's the same data in each of these columns, just rearranged differently. Some of these artifacts you really get emphasized out. It really kind of shows you more of these interesting patterns and things that you didn't think were going to happen are happening here. So with this, I mean, how does that GSR pipeline look like in these transform plots? I mean, I'm talking about GSR here, but this is really useful for essentially any type of pre-processing pipeline you can think of. It's really the tool that I think you should be using to analyze your data, or your pipeline, I should say, as well. So here's an example of that subject. In a random ordering, you don't see much globally or much WSDs. You just see kind of a sort of flattish profile with some evidence of essentially some of these WSDs which are correlated to these movement events. If I apply GSR, I don't see much there, and of course remove it all there. But if I look at the GSR but reordered, all of a sudden these things which we thought were gone were actually still there, and these red sort of arrows there. And more impressively or depressingly, if you look at the cluster ordering, all of a sudden now these things really pop up and they're really emphasized. So it looks like the GSR didn't really work as you thought it would. You still have these big residual noisy artifacts. So if I now look back at the three different categories of subjects, if you look at the random ordering, it looks like you've done a really, really good job. If I look for global signal ordering, all of a sudden, there is some stuff that's still residual. But what's really quite interesting and we'll see is that the single phase WSD, so everything that's correlated in the same sign, seems to be removed. Just some little bit residual stuff here. But of course, in the cluster ordering, what really gets emphasized are these patterns that seem to be out of phase. You can see here that in these blue arrows, these things really pop up, and they really have not been removed. And it's different depending on which category of subjects which I've made comes up. And so, in summary for this little part here, is that it's not that striking under these plots. It's essentially the same information but different orderings. And the reason why in these multiple phases it still pops up is because when you take the average of, course, all these voxels, essentially the positive negatives, they cancel out. And so the global signal, or the barometer signal, is not representative of this sort of pattern. And so this is like brings forward a new way, new way of thinking to get rid of all these patterns. But I wanted to take a step from that, and I guess the key thing I want to just show you are these reports. And so essentially, all this can be done quite quickly with a pipeline which we developed called DICER, which exists on GitHub. This works on Massive. We tested multiple times, and you can get all your data sets to whatever type of pre-processing you want. So we also use other pre-processing pipelines as well, and you'll generate a huge HTML report which has every subject. You can click through every subject and just check up individually how it looks like. I think this is a really nice way if you're looking at large-scale multiple data. I think it's most data you'll be working with when you use Massive will be large scale. So you need a way to really understand and check with them quickly. And this is the pipeline we have got, which does this. We also have other things which I'll talk about in a second. But yeah, I think this is the way you should really be thinking about when you look at the data. You need some kind of summary metrics for every type of pre-processing pipeline you look at in your data sets as well. So I mean, I'll talk about this briefly because I think it's not the most important part of the talk. But what we did is we essentially found a way to remove those residual parts. So we found that these anti-correlated parts still exist. So this is a new technique to correct this out, mainly spearheaded by Ben Fulcher, where instead of looking at taking the mean, we looked at weakly correlated structures, and we use this clustering called dbScan, which is like another thing like k-means, a different thing. And you could determine parts that are weakly correlated together. That's a regional target of weakly correlated diffuse widespread deflections. And from that, you can essentially develop new regressors that really take into account this sign flipping. I just want to show you quickly that we did this, and it looks how we estimate more regressors, and we can really remove these in the cluster ordering techniques, these top 3 panels. If you use DICER, you kind of remove all these large scale activities, or the widespread deflections, and you're left with a CETA which looks flat. I mean, it looks like there's nothing there, but we've applied multiple techniques to really test whether or not we've removed too much. And it turns out we haven't. We actually improve our statistics on a whole bunch of things. Let's go there. So I will say, in summary, it's like a QC talk. But the random ordering, how you order your voxels when you do this large scale analysis, can be deceptive. When you transform the voxel ordering, there can be more than meets the eye. So you should interpret them with caution. We've developed a new technique which can possibly remove all these things. We replicated these on 3 independent data sets, open source data sets. So again, with Oversize framework, Cambridge multi-echo data, also with Beijing Zhang from the F1000 Connectome project. Also, I have this paper link here. This paper link was quite hard to get through review because at one point they just said, okay, this is all fantastic. Can you now replicate it on the HCP and their task data? And this would have been quite daunting, but again, the magic of Massive, this is done in a month instead of a year, and we could port everything from Massive and use the technique quite nicely on these HCP data sets. So I just want to thank all the people involved, Ben Fulcher, Lyndon Parks, who's at UPenn at the moment, Christina Savarita, and Alex Juanito. And yeah, thank you.
Fantastic. Thank you so much for this great talk. Yeah, we're doing very well in time. So we move on to the last talk in this CDL at M3 session, which is Dr. Yu-Chin Chen from Monash University. So looking forward to that talk.
Let me share screen. Sorry, this one. Okay. So.
Fantastic,
So
that comes through well. Perfect.
So I'm going to talk about some of my PhD work, which is characterizing scale-specific cortical shape asymmetries. If we look at these 2 images, we can see they are a rabbit and an elephant. If we add some noises on them, we still can understand this is a rabbit and this is an elephant. But if we look at very tiny scales, for example, here or here, we can't see the big pictures of these 2 images. But for the current neural image analysis paradigm, people tend to focus on finer and finer resolutions, but may ignore some interesting information at very broad scales. And this is what our scale-specific analysis for. Specifically, for example, if this is a region of interest, and if this is another region of interest, and if we set the statistical threshold here, then this point is significant. If we set the threshold here, then these 2 regions are significant. If the threshold is here, then all of them are significant, but we still can't see the effect, the underlying effect across all of these regions, which is this black line. So this is why we would like to use the scales-based analysis. Before talking about the geometry decomposition, I'd like to talk about the Fourier decomposition, which is the most classical example for signal analysis. The red line here is the original wave signal, and we can decompose it into the waves at different frequencies. Each has its own frequency, and we can also calculate the contribution of each decomposed wave signal to the original wave signal. Similarly, we can use the same approach for decomposing the morphological features. In our study, we use the Helmholtz equation, which is an ubiquitous equation for describing the diffusion process, like the heat diffusion or electricity diffusion. And here I use the cortical thickness as an example. This is the original thickness map, and we can decompose it into the spatial scales at different scale, from very close scale to the finer scales. And then we can also calculate the contribution of each scale to the original thickness map. And each scale has its own corresponding frequencies. Here we call it as the eigenvalue. In our study, we focus on the shape of the cortex. This is because if we look at these three objects, it's clear that they have the same volume, but their shapes are different. And previous studies also found that shape is more sensitive than other size-related index like cortical thickness, surface area, or volume. And in our study, we calculated the differences between the eigenvalues from the left hemisphere and those from the right hemisphere to characterize the cortical shape asymmetry. And we use the three open source longitudinal data sets, which are OC3, ETNI, and HCP for the analysis. And first, if we look at these four matrices, they are the Pearson's correlation between the shape asymmetries at different time points. And the diagonal line here is the correlation between the same subjects. And the off-diagonal elements here are the correlation between the different subjects. And we characterize the identifiability scores to represent the subject identifiability. If the score is higher, it means the better identifiability. Interestingly, we found the best subject identifiability is shown as the coarse scales, which is this one. And people tend to focus on very fine scales, which is like here, and the identifiability is not as good as the coarse scale. We also compared the subject identifiability of the shape asymmetry with functional connectome fingerprint. And this black line is the identifiability of the shape asymmetry from the HCP data. And these colored lines are functional connectivities at different resolutions. No matter what the resolution scales are, our shape asymmetries have the better identifiability than all the functional connectome fingerprint. We first analyze the shape asymmetries between females and males. And in these 5 scales, females and males are significantly different. We also used the HCP data to analyze the heritability. We found the heritability of the individual hemispheres at very close scales are high, but the heritability declined at the final scales. But for the shape asymmetry, the heritabilities are generally very small. Instead, subject-specific environmental effects strongly affect the shape asymmetries across all the scales. In another studies, we used the HCP early psychosis datasets, and we found between these 2 scales, the healthy control and early psychosis are significantly different between the shape asymmetry. And we also use the CCA analysis to find that the shape asymmetry is significantly correlated with their symptoms. In conclusion, we found shape asymmetry is a highly visualized feature and is mainly driven by the environmental effect. And shape asymmetry has a high potential to analyze abnormal behaviors and symptoms. And I'd like to thank to my supervisors, Alex Fornido, Kevin Aquino, James Penn, and also my colleagues, Arena, Ashley, Sid, Chow, Eugene, and all of the NBS lab members. And I'd like to thank to Macy for us to store and analyze the data. Thank you.
Cool. Thank you very much for this great talk. So we have a quick Q&A session now for this block. So if you just have questions, post them in the chat, and then all of the speakers can maybe comment on them, and then we move on to the next block. So I guess we have a couple of minutes for this. Also, if you have any other questions about CVL data collections, please. Okay, then I don't see questions yet. I'll start with a quick one. Would you would you say that CVL and the data collections that made it available made your work easier or possible? So do you think it's just something that you could also do on a normal computer, or would you say it's really something that's crucial to have?
It's for anyone, right?
Yeah, exactly. Yeah, just to get the discussion started. Yes.
I would say, I mean, I talk about a bit in my talk that it would not be possible to do single computer type of thing, and definitely needed a cluster. So, I mean, I didn't really make a point of this, but, you know, we had multiple data sets in our work with multiple pipelines, multiple variations. This would have taken, you know, way too long. I've been working with similar clusters in smaller scale. So we actually needed a cluster, but not only a cluster, but a big cluster to do these, and, you know, running parallelization of, you know, a few hundred jobs, multiple iterations of these. It took a long time on the cluster anyway. So doing it not on the cluster would have taken a very, very long time. So 100%, it was very crucial for my work.
Yeah, cool. Okay, that's good to hear. Yes, and also, yeah, see a lot of these examples, what you can actually do then with this. Yeah, cool. Do we have any questions in the chat now? I don't see them. Q&A. Okay, now there's one. Eco asks, can the signal extracted by DICER be correlated to ICA components? I think that's a question for you, Kevin.
Yes. Well, thank you for that. In terms of ICA components, I mean, I would assume you mean ICA fix components or any type of ICA components. In reality, we did check these, and they're not exactly the same ones. I mean, I guess what we checked really was whether or not the correlations to these components were related to ICA networks. I mean, that's the way we really tested this. And on the whole, no, they weren't really. There were some cases where various parameter tunings can be correlated. That's required us to really test this out. Also, that's another way that people do to check, so not just DICER, but any other progressing pipeline, whether or not your presentations of these regressors are correlated to any functional network. That's a big thing you should always test. And in some instances, yes, but on the whole group level, no.
Cool. Yeah, cool. Thank you for this interesting question. And yeah, so I don't see anything else in the question and answer, and also I don't see anything in the chat. Well, then I think since we're doing very well in time, let's move on to the next block. So the next block is like basically other big data collections. And here we have two speakers. So we'll start with Eli Müller from University of Sydney. So let's get started with brain state kinematics.
Cool. How you going, guys? Thanks for having me. Just gonna share my screen. All right, we cool?
Yep, that's perfect.
And, that. All right, this is going to be a bit of a whirlwind, so strap yourselves in, but here we go. The title of the talk's Brain State Trajectories and a Story of Open Data. So the common thing that runs through a lot of projects I address here is the usage of the Human Connectome Project dataset. As far as like how that data has been curated for me, I'm a little bit detached from it. I work in a group, and in fact, my collaborator Max Shine had done a lot of the pre-processing on the Human Connectome Project data before I got it. But this is really a demonstration of the power of having an open data set that's always at your disposal, and how it can sort of link together projects and allow interpretations that span projects. So just a couple of definitions before we start. A brain state, a snapshot of the brain's dynamics that can have properties of interest or properties that are available to the observer. And obviously the brain state trajectories are how these brain states change in time. And a key sort of question of this project is there something in those trajectories of brain states that can tell us something about learning, disease, different types of tasks. So yeah. Also, first and foremost, a low dimensional system. Well, correlations exist in the brain. Like neurons have a level of independence, but at large scales, we find these persistent correlations that sort of hang around, and that at its core defines a low dimensional system. The idea that there are lots of degrees of freedom that the system has, but it only exploits a few of them at certain times. And that basically, like that low dimensionality is what underpins persistent spatiotemporal patterns. So a key question that might arise from this, and in fact, it's been a key question of neuroscience and neuroimaging for quite some time, is there some sort of useful basis that's sensitive to these patterns, be they resting state networks, parcellation schemes, and indeed like principal components, which is a nice method from applied mathematics. So what is principal component analysis? For those that don't know, it's just an eigenmode decomposition technique that's designed to sort of, you can think of it like rotating your data set around such that you can see the most variance along the axis that you had some X and Y in the two-dimensional case, and you want to do some sort of rotation so that the spread is maximized. It's depicted in this diagram at the bottom here. All right, with that in mind, so I'm going to talk a little bit about some work here that I wasn't involved in by Shine et al. In Nature Neuroscience 2019, where he took the Human Connectome Project data and in all its glory, which is a lot of subjects recorded across a battery of cognitive tasks, sort of grouped along the top there into emotion, gambling, language, motor, and impact, and so on. And he concatenated all those time series, and then he did a principal component analysis to it. And what he identified was 5 sort of, I don't know what you want to call them, like principal brain maps, principal components that constitute a brain map. They're like eigenvectors of the covariance matrix, for those who like math. And you can basically track how much of each of these spatial patterns that you have on the left here in time, at particular instances of time. And you can see that it turns your time series data into something that's a little bit more interpretable, and that you can see clear distinctions between some of these cognitive tasks. And that's indicative of them recruiting or unloading from certain brain regions. And indeed, this can be viewed from the cortex, from the cerebellum, and the thalamus. And if you look at average trajectories of subjects across these cognitive tasks, you can see that there's some sort of persistent low dimensional trajectory that goes on. It is as the subjects move to unique parts of this low dimensional state space. They're recruiting certain brain regions in some instances and not in others. And in fact, here's another way of, even the first 2 principal components can disassociate unique brain configurations to both memory, language, motor, and cognitive tasks relative to a null distribution. So a follow-up study of Shine et al. In Neuron 2019 was to take those same principal components, so that same basis set that's been honed on the cognitive tasks I just mentioned, and apply it to a new data set. In this case, subjects were in the scanner and they were doing something akin to it's called the Latin squares task, but something akin to a Sudoku game. It's more based on shapes. And the complexity can be varied here, binary, simple, and all that to quaternary, which is quite difficult. And you can go through and take, in this case, the 3 principal components that have been defined on the Human Connectome Project data and track subjects' trajectories as they perform this task. And the authors showed that there was a clear distinction between subjects who were making mistakes and getting errors and those who were correct. So there was a low dimensional signature of the subject performance. But a key question that came from the study was, are there methods that go beyond variability? In this case, we were looking predominantly at principal components, which are defined around variability in your time series. So we pivoted. This is a study I was involved in, where this is a new task now. So it was a learning task, and the paradigm is essentially subjects are in the scanner. They're shown this instruction screen on the left here. And they're to associate 2 patterns with the left hand button press and 2 patterns with the right hand button press. And then they go on to a practice phase where they're just showing a single pattern and they're to recall what the association was, and then respond correctly or incorrectly. So a key part of carving this task up is sort of quantifying what your question is. And for us, it was really, is there differences similar to the previous task I mentioned, where there was some low dimensional difference between error and correct? Is there some low dimensional difference between what people are doing early on in this task, the first few times they're trying to remember what the instruction phase told them, and later on, so that's the early late axis here, and then the slow and the fast. So some subjects responded really quickly, it was straightforward for them, and then some much slower. So a little diagram here is sort of, I won't go into the method that we used here, but it's another linear algebra technique. Principal component analysis is depicted by this arrow here, much like the previous figure that I showed. But there's another method called linear discriminant analysis, which basically goes through and tries to separate. If you define two clusters within your data, it'll say, well, if you rotate it this way, this is the direction you should look at your data set such that I can see the greatest difference between the two means. And in fact, it also can ensures that, in this case, the ovals don't overlap. So the covariance is unique, are still maximized. And we applied linear discriminant analysis to this data. And you can see in the top two panels are just taking if you've got 4 classes and you use this technique, linear discriminant analysis, you end up with 3 eigenvectors. And I've put a scatter plot on here. So each dot is a subject doing one single repetition. But they had 8 repetitions. So they had an instruction phase, and then they would correctly identify a pattern up to 8 times. And you can see that the repetition is colored here, blue through red. So subjects are moving early on in the task, they're in this blue region, and then as the task progresses, they move into this red region. So there's some sort of low dimensional representation of repetition in the task from this method. And then equally, well, reaction time is depicted here, but it's a little bit more scrambled and not intuitive. But if we go through and look at what each of these eigenvectors has a brain map, just like the Nature Neuroscience work from Mac. And I've depicted the 3 brain maps that correspond to those eigenvectors down the bottom here, and they're loading across repetition. Think of repetition like time in the task. And you can see here there's clear movement on the first eigenvector as we recruit more of the visual and medial frontal regions. Eigenmap 2 has this sort of intermediate loading and then unloading again. And this has got some dorsolateral prefrontal cortex and some late ventral visual activation. And then finally, you've got something that's, although it does have an inverted U shape, something that's more of like a convergence. You start in some particular part of state space and then you roll down and settle into some new minimum. And here we've got like a large temporal lobe activation. You can take those vectors and define things like a task manifold, which is showing how you want to move across the task left to right, and then you want to move from blue to yellow as you get better at the task. So if you're fast, and at each sort of phase it's depicted here, you can show the slow performers, the fast performers, and the improvers as a low dimensional trajectory here as well. And indeed, I've resolved those on the bottom there. So you don't have to look at the 3D plot just as a one-dimensional slice. All right, and then somewhat of a pivot. But still with trajectories and open data at its core, like trajectories are constrained by anatomy. And there's some great work by Shine and Progneuro 2020 that carved up the thalamus into these 2 distinct connectivity types, which is called the core and the matrix. Long story short, the core are those canonical targeted thalamic projections to the cortex that are involved in early visual processing. Mind you, the matrix are as well, but the matrix are distinct in that they project to the cortex in a very diffuse manner. They send their connections, their projections quite broadly in a spatial sense. And there's some cool work sort of carving that up. So we took that anatomical principle and decided to build a model of it that included a neural mass model. Again, I won't go into the details here. The key thing to remember is we just wanted to model those local connectivity and diffuse connectivity. And when we did that, what came out was this nice intuition from physics, which is you've got some type of attractors. Attractors are like a low dimensional property of the system where, like, you can imagine the brain like a marble rolling around on a lumpy manifold. Sometimes it sits in little wells and gets enough, sometimes it gets enough energy and rolls over peaks into other wells. And in fact, turning up those diffuse connections is very much like flattening the wells of your system. Your marble was in a deep hole, you flattened that hole, and now the marble has enough energy to roll out of it quite straightforwardly. And we're using that model, we showed that matrix-like connectivity could facilitate something like that. And when we took the outputs of the model and we said, all right, well, let's revisit the Human Connectome Project data and see if we can contrast rest and task, because Shine had this theory about task and the arousal system recruiting this type of diffuse connectivity. So we basically gave a model fitting algorithm that 1 degree of freedom, the diffuse connectivity, and said, all right, I want you to fit the model outputs to the Human Connectome Project data outputs in the task and rest conditions and tell us how much of diffuse and non-diffuse connectivity you had. And lo and behold, that's what's shown on the top right here. So the probability of all subjects and where they sat in the state space. And basically the result of this was that task was recruiting more these diffuse connections, which was consistent with this arousal-like signal, and some of the theories from Shine's earlier work. And hopefully I'm on time. Sorry if that was a bit of a whirlwind, but I guess the summary here is that open data, well, I'm a physicist, so for me, the idea of open data is awesome because it allows results to be compared across projects and indeed contextualize them to the results of previous work that have used the same data. And in fact, I maintain, a library of open data sets, and every time I have a new idea or a new method, or I'm just curious how certain ideas link together, I will revisit some of these open data sets. So it's quick tests of new hypothesis, contextualizing your results in the context of other results, and obviously to compare novel methods, because I'm sure we all in neuroscience know that there's so many ways to skin a cat for neuroscientists, and there's just way too many methods and not enough people synthesizing what the hell's going on. So, and yeah, that's it. Thanks, special thanks to Dr. Max Shine and Dr. Brandon Munn.
Thank you very much. Yeah, that was perfect in time. And we even have a little bit of time left if there are any questions. Just post them in the chat. If there's nothing, I think we can also move on and then keep them for after that.
Sounds good.
Cool. So then the next talk, Aaron Boren from University of Melbourne. That will be the last one for this block, and looking forward to this talk.
Thanks, Stefan. Can you see my screen?
Yes, we can, and we can also hear you clearly. Perfect.
Great. One thing. Yeah, so thanks to the organisers for this excellent workshop. So I'm a postdoc working across the University of Melbourne and Royal Children's Hospital. And my talk is going to be, I guess, a bit more clinically focused on the kinds of applications you can do with open access data sets. And specifically, I'm going to talk about some work we've been doing using the Human Connectome project to optimize a new treatment called deep brain stimulation for people with a severe kind of epilepsy known as Lennox-Gastaut syndrome. So what is Lennox-Gastaut syndrome, or LGS? So LGS is really the most severe form that epilepsy can take. The onset occurs in young children who have very frequent seizures, often hundreds per day. And one of the most defining features is tonic seizures. And I'm going to show you a little video of what one of these seizures looks like. So what you can see is a kind of sudden contraction of the child's muscles, which cause the limbs to rise upwards and off the bed. So these are a particularly nasty form of seizure, as they can occur suddenly while the patient is upright, and this often results in traumatic physical injuries. So more than 90% of these patients don't respond to standard medications. Around 10% die during childhood, and almost all develop severe intellectual disability. So this is really a group where new treatment options are urgently needed. However, until recently, the mechanisms of seizures were unknown, which has prevented treatments from developing. So one of the most promising methods to try and understand these mechanisms is combined EEG with functional MRI. And this allows us to visualize whole brain activity at the time of epileptic discharges that we can capture on scalp EEG. Suffice to say that EEG fMRI is not straightforward to acquire in patients with LGS due to their severe cognitive impairment. However, in recent years, we've been testing different strategies to capture this data. So in one study, we acquired EEG fMRI in a group of adults who were scanned after some really quite talented radiographers encouraged them to fall asleep. And in a separate group of children, we acquired scans under a very light form of anesthesia. In both groups, what we saw was this really widespread, highly desegregated activation during epileptic activity, particularly in frontal and parietal hub-like areas, as well as subcortical structures, including the brainstem. And these results really speak to the severe generalized nature of this epilepsy, And they suggested that any treatment may need to be capable of modulating abnormal excitability of these large and distributed networks. So this led to the concept of modulating the epileptic network of LGS using a technology known as deep brain stimulation. So this has been used for many decades in other conditions, most famously Parkinson's disease. However, it's only recently been adopted as a potential epilepsy treatment. So the procedure involves implantation of electrodes into deep brain structures, and these are connected to a battery pack which delivers programmed patterns of stimulation. One of the key parts of DBS is choosing the right target. So in the thalamus, there's a nucleus called the centromedian nucleus, or CM, which has several interesting features that make it a potentially attractive target for this kind of epilepsy. One of these features is an apparent ability to modulate diffuse cortical excitability. So the EEG trace you see here on the right shows scalp recorded changes during low frequency stimulation of the CM. And what we see is a diffuse cortical response, particularly over frontal channels, which in many ways mimics what we see during seizures of LGS. So this suggests the CM nucleus may be an effective way to tap into these broader seizure networks. So armed with this knowledge, we recently embarked on the first randomized clinical trial of centromedian stimulation for LGS, which we call the ESTEL trial for short. Very briefly, this involved 20 patients who underwent bilateral implantation and were followed before and during stimulation using detailed clinical measures. The efficacy results will soon be published. However, today I'm going to focus on a post-trial analysis where we aimed to test the association between patient-specific locations of stimulation, the connectivity of these locations, and seizure outcomes. So after 3 months of active stimulation, there was a median reduction of seizures of around 50% relative to baseline. However, there was a bit of a range from around 20 to more than 80% reduction. One of the first things we asked was whether efficacy might be associated with standard clinical information. And we tested this for a range of variables, but didn't find any obvious associations. To take just 2 examples, outcomes didn't appear to be associated with the patient's age, nor their level of disability. So next we asked whether outcomes might be associated with the locations of stimulation. So to test this, we first began with patients' pre-surgical MRI, and this was merged with a post-surgical CT scan, which shows electrode positions within the thalamus, allowing us to determine the locations of each stimulation contact. So this is a model of each electrode showing 4 contacts. The choice of contact and the stimulation amplitude varied across patients. And these patient-specific settings were used to estimate a field of stimulation using a really nice method published by Andreas Horn. From these, we defined an area where stimulation likely had influence, and these areas are called volumes of tissue activated, or VTAs. So going from this 3D view to a 2D coronal slice, we can see that these VTAs allow us to define the zone of stimulation in a common MRI space. Given subtle differences in surgical positioning, the exact zone differed from patient to patient. And so by combining all patients, we can define a heat map showing overlaps, which allow us to perform voxel-wise statistics. So using this method, we asked which zones of stimulation yielded better outcomes. So the warmer colors here indicate areas where stimulation was more effective and the cooler areas where it was less effective. So on these coronal and sagittal views, you can see that stimulation was most effective inferiorly, laterally, and anteriorly. Displaying the results in 3D, again in a coronal view, we can appreciate the relationship to nearby thalamic nuclei, with stimulation appearing to be most effective in the lateral portion of the centromedian nucleus, near where it borders the ventrolateral nucleus. So next we asked whether connectivity of these stimulation locations might also be important. Very briefly, this began with each patient's VTAs, and these were input to a diffusion MRI structural connectivity analysis, which we performed using up-to-date methods in MRTrix. And this yielded a connection strength between each VTA and all other brain areas. And so finally, at each brain area, we then just correlated connectivity with patient seizure reduction. So one wrinkle to this analysis was that we didn't have pre-surgical diffusion scans for the complete cohort, in many cases because high field scanning was contraindicated due to patients already having a variety of other medical devices implanted, such as vagal nerve stimulators. And this is where we turned to open access data for help. So in particular, we used the high quality control scans from the HCP, and then to see whether the results were reproducible using more clinical quality data, we repeated the analysis using a cohort of patients who participated in some of our previous imaging studies of LGS. So when we looked at where connectivity was greatest, we saw a pattern consistent with the known projections of the centromedian nucleus, including dominant connectivity with primary motor and pericentral cortex. And this was similar across the normative and quote unquote diseased matched data sets. So just to clarify, these results show where there was connectivity, but not which connections were associated with clinical outcomes. When we performed this correlation, a kind of different pattern seemed to emerge. So seizure reduction was most positively correlated with connectivity to premotor and caudal prefrontal cortex, and subcortically with the putamen, brainstem, and cerebellum. And once again, the findings were pretty similar across the two data sets. So we noted that this kind of frontal pattern showed many similarities to these EEG fMRI maps you saw earlier, showing areas of epileptic activation. So in a final analysis, we tested whether the strength of connectivity to these seizure-related areas might be associated with DBS efficacy. And indeed, in both analyses, we found a moderately positive association between seizure reduction and connectivity to these areas of a priori EEG fMRI activation. And so this kind of adds weight to the hypothesis that efficacy of DBS really depends on the degree to which our stimulation target is connected to a broader epileptic network. So there are three take-home messages from this work. Standard clinical information, such as baseline disability, was poorly correlated with seizure outcomes, at least within our modest sample size here. We think this speaks instead to the importance of parameters surrounding stimulation itself, including the specific location. There seems to be a kind of sweet spot for DBS in the anterior and inferolateral centromedian nucleus of the thalamus, and this will be important going forward for planning future trials. And lastly, connectivity analyses using open access data really allowed us to ask clinical questions which otherwise wouldn't have been possible given the significant obstacles involved in acquiring advanced MRI in this population. And so this showed that connections to remote areas known to drive seizures were most associated with benefit, particularly frontal and premotor cortex, the putamen, and brainstem. So we think this is exciting because it lays the groundwork for adjusting stimulation based on these patterns of connectivity. And this is something we're now starting to explore in a follow-up trial where we're seeking to test whether therapeutic benefits can be further enhanced by targeting stimulation to locations with greater connectivity to the areas we identified in this analysis. So I hope that gives you some idea of the ways that open access data can have some real world impacts. And I just want to thank my clinical collaborators, supervisors, and especially the patients and families who made this work possible. Thanks.
Fantastic. Thank you very much. Yeah, we have a couple of minutes before we head into the break and post questions in the chat. And we already have one, which is for Dr. Eli Müller. And the question is, could you explain what you meant with that the PCA is relying on representing data variability and how other methods can be used to capitalize on other properties of the data?
Yeah, I'd like to give a shout out to the question poser, Max Shine there, who happens to be my boss. Yeah, sure. So principal component analysis somewhat how I was iterating something along the lines at the start, which is that you have some sort of data that has some, is in some basis, whatever it may be, however many dimensions you've got, 400 if you've got some 400 parcellation of the brain. So there are some dimensions to it. And when you do principal component analysis, essentially what you're doing is rotating that frame of reference so that you're looking at it from a new perspective now, and you can see the largest amount of variance, the largest amount of spread of your data. And that's a piecewise problem, because you will end up with 400 principal components if you had 400 observations. But now, if I grab the first principal component, it contains the most variance in my data. And then the second bit, the second most, and so on and so forth. So it's a means to capture, if you want to think of variability as information, it's the means to capture the most information in your data in a low dimensional sense. So the first 10 principal components will carry more information about the system than just 10 random regions grabbed from the brain, right? So that's the idea behind the principal component analysis. But sure, you can use other methods that have a different optimization goal. I'm quite fond of one called dynamic mode decomposition, which looks at how the state changes from time point to time point and tries to find a mode. Like you can build, basically it's all linear algebra. That's what, you don't realize this when you're doing mathematics in first year in uni, but your life becomes linear algebra the longer you stay in research. So yeah, you find this matrix, you find a matrix like an autoregressive first order matrix or something like that, and you can do eigenmode decomposition. And again, all you're doing is rotating your data around or reformulating it into a new basis set that either maximizes or minimizes some objective function. And principal component analysis, it's variance, and dynamic mode decomposition, it's the amount of predictability you can get from time point to time point. But there's so many different ways of doing this. I mean, yeah, dimensionality reduction, I think, is just an awesome technique in neuroscience in general. And depending on your question of interest, you shouldn't just go in and throw random methods at things and see if things pop out, but they can be very powerful if they're motivated by hypotheses. So, and do you think about, variability? Do you think about dynamic modes? Do you think about correlation clusters? Whatever you, yeah. So that's a bit of a loaded question to carry on, but I'll stop there.
Okay, cool. Thank you very much. Cool. We have another question, this time for Aaron, from Marcus Bart. Now that you have a better understanding about which subpart of the CM has best outcomes, if I understood this correctly, now how can you accurately place the DBS electrode?
Yeah, really good question. So we did do a lot of work kind of in the lead up to the trial to sort of try and acquire various sort of MRI scans that kind of allowed us to better visualize internal thalamic architecture. We had to sort of take a very pragmatic approach to it, partly because these patients, many of them, they can't be scanned at sort of, very high field strength MRI like 7T and so on, which I know sort of, other groups are starting to use to sort of, get finer and finer resolutions to visualize thalamic nuclei. So we had sort of ended up compromising and acquiring something called MP2 RAGE, which is sort of just a kind of a development upon T1 MP RAGE. And we found that it sort of did provide reasonable visualization of internal thalamic architecture, at least enough to sort of be able to sort of see landmarks nearby the centromedian nucleus that you can kind of then target from. But in terms of sort of targeting at the sub-nucleus level, like sub-centromedian nucleus, I don't know that there's any MRI techniques available at the moment that will allow that. Some alternative ways are kind of based on electrophysiology. So intraoperatively, we can acquire microelectrode recordings along the sort of trajectory of the electrode lead. And they allow us to sort of try and figure out, are there sort of electrophysiological markers of where you might be in the thalamus during surgery? And so we've kind of acquired that data, and we're currently sort of trying to unpack if there's any signals in it that might allow us to see if we're in one part of the nucleus or another. But really, I think that's going to require much kind of larger data sets to sort of properly disentangle.
Fantastic. Cool. Thank you very much. Okay, with this, we planned now for a little break because this was a lot of input. And before we start with the break, I just want to acknowledge all the work that Chao, Catherine, Farnoosh put into this webinar. So they actually invited all the speakers, got up with everything, and also prepared the next session, the hands-on session, where you now see a little bit what CVL can do to make a lot of this happen that you saw today in the talks. And with this, we planned 5 minutes. So let's come back in 5 minutes. and then that the hands-on session will be a preview of what you can get if you sign up for this interactive session. And I think Catherine will also post up the link again if you haven't signed up yet. I hope there's still spots left, so we'll see. Okay, see you back in 5 minutes.
Yep.
Yes,
Cool.
I am.
Okay.
I'm not going to do too much talking. All I wanted to say was thanks everyone so much for participating in the poll. Nearly all of you have, and I'll just give you a quick result there. I might just end the poll unless anyone's desperate to participate. But the result is pretty much 49 to 51% of you. So 49% of you have been working with open data, which is amazing. For those of you that haven't, hopefully you'll get some tips and some pointers and advice about how to go about starting that process through Chow's talk and through the other presentations we've had today. Now, all I was going to do was, I'll end the poll, and I will just very briefly, just to give you a quick gratuitous advertisement for Chow's fantastic session that he'll be hosting next week. And it's going to be a workshop on exactly how we go about doing these things. So I am just going to share my screen with you. There is a dog barking a lot in the background, which is quite annoying, isn't it? It's always these things happen. All right. So if I just gratuitously share this presentation, which will not be very long at all. So for those of you who haven't met me before, I'm the Training and Community Engagement Manager for the ACCS project. So I help to put together the events and make sure that you're registered and all up to date with getting your access onto CVL, which is the Characterization Virtual Laboratory. Now that's the environment that Chow will be working with during the workshop and where the data collections are. For those of you who might be coming from Queensland or for other locations around Australia, I just want to give you a quick heads up that this workshop will be running completely out of Monash because it has the collections that Chow will be demonstrating and working with. And if you require more information about that, please do get in touch and I can give you a hand. So just very quickly, here is the link to register. Please do. And I will also put that again in the chat, and the information will be available on the OSF as well. So please register here like so. And just to cover again what you'll be learning when you come to the workshop, and that will be learning about the types of data that are in the collection, that are in the CVL itself, and the mechanisms and pathways that you'll go down in order to gain access to them. We've seen a lot of the talks this morning about what you can do with these collections, but Chow will also be going into some practical steps on how you can work with them and what you can do. Sorry?
Some basic, yeah, that's it. Yeah, some basic things like just view the data, let's go through it, what it looks like. Yeah. In the next session, yeah.
So hopefully we'll get you in the environment, you'll get a look and a feel for how that operates in practice. And so I just want to reaffirm that you've got to have access to the CVL at Massive in order to participate in this workshop. If you do not have an account, please email me today or tomorrow. If there's a number of you, I can host a session for us all together online on Zoom in order to get that set up. But if you do have an account, or maybe you have an account with Weena or something like that, get in touch with me too and we'll sort it out. So if you need some assistance with that, you can also follow this link here, which will give you some more information about how to set up that account, or I'm happy to meet with you later in the week and we can do that together. And the only other thing I would say is if you do have an existing account with CVL at M3, please do make a request to join this particular project here, OH21. If you've joined us before for a workshop, you'll be familiar with this project. I have cleaned it out, so I would ask you again to please rejoin that project if you haven't already for this workshop. So at the end of each workshop, I will just be clearing that out to make some more space for each participant. So if you could please make that request over the next week, I will email you if you've registered and we can work on that together. If you have not registered, again, here is my gratuitous cat gift because everyone loves a cat gift. And please follow the link and register. And that's pretty much all I have to say. And Zhao, I'll get out of your way and leave it all to you now.
Thanks, Catherine. Let me share my screen first. Okay. Can people see my screen? Okay. Okay.
Yes, all working perfect.
Yeah, thanks. Thanks, Catherine, and thanks everyone's great talk, especially the researchers talking about those great applications about using the open access data. So emphasis for Kevin, Yuqi, and Stuart, so who is working on Monash, working with Massive before then. Today I'm going to show you and walk through with you the HCP data, and then the Massive environment will be exactly that, what they're doing, their applications and projects is exactly the same environment. So today, I think I'm going to just roughly separate it into a few parts. So just what is HCP data, and then basically then how to access to HCP data, and I'll emphasize like how to access HCP on CVL and Massive. And we're going to take you through, have a look at what the data looks like in there. Lots of people who is actually haven't used that before or don't have the experience, want to know what it looks like. And then from this session and also the hands-on sessions, I'm going to quickly show how to use like FSIs and the Connectome Workbench. Just do some visualization and then looking at the data, etc. Okay, so we start with the HCP data. I actually know HCP data so probably around 10 years ago. So when I about to finish my PhD, and I still remember my co-supervisor saying that was the game changers of the neuroimaging field. Because around that time, the data sharing is not, it's a kind of a new thing. It's not easy to sharing data. But as time goes by, the project is going well, and we see the potential, and we see how this is helping reshape the research. And then HCP itself is also going upgrade to this Connectome coordinated facility. So it's not at the beginning, the original, the young adult, HCP young adult, they're starting from like 500 and 900, now it's 1200. But they also have the other stream, like the lifespan HCP data, so which is have a more age range. Say this is the aging one to across 100 years old, and so the development focus on the young adult. They even have the HCP baby, which is the even younger brains, and also they have the connectomes to disease, which is have like dementias or younger earlier psychosis or the other disease. As Farnoosh pointed, some of the data are already available on CVL Massive. Depends on the accessibility, so we can get you access to the different levels of the data. Okay, so how to access data? So there are mainly 3 ways to do it. Firstly, it's the easiest just to download using Connectome database, just on your browsers or anywhere, you can download it. And second is Connectome in a box, which is a bunch of hard drive you can order. And then the most easiest one, if you are on Massive, is just a CVL at Massive, you can access to the data directly. We start from the beginning. So everything that the download data from the Connectome database is simple. Just log on to the db.humanconnectome.org, and you will come to these browsers, and then you just do register it and get your account set up. Once you approve, and you will have this interface. Depends on how much data you can access. So you can, at least you can access this 1200 subjects of data here. With this interface, so you can firstly use this pop-down menus to explore the subjects. Say, for example, if I just interested in the people who have a 3T MRI scans, and then you just click that, and they give you a summaries of all the people that are having this group that people having the same data set that you selected. And you got a full list of all the participants here, and you got information of release. Remember, that's starting from the very beginning. They have a Q1, Q2, Q3, S500, S900, and S1200, which is the final one. Another thing is you can see the ages by the age range. So which means that they just at the very beginning, once you get the access to the restricted data, you have more information of the behavior or demographic of all of the data that you want. Probably people's doing some data management, you know, this interface is very similar as XNet. So that's right, XNet is powered by HCP database behind. And then so if we click, if you know XNet, so after you click on the participant, you go to the next level, which is the modality of the different scans. And you can click on those different modality. They give you the details of the metadata of scanning protocol, etc. Those information, you can get it easily from here. And if you click on here to get files, you get another pop-up windows. Then you can select all of these files. And the one you like, there's a different things, the unprocessed, preprocessed. Choose the one you like, and then just click download straight away. So that's one way to download. Also, if you want to download the more systematically, you go to this download the image, the pop-down menus, and then again choose the group that you want, and then you come into this. You will come into this download interface. Again, you can select what type of the data you want. They have unprocessed, preprocessed, and analysis. And on this side, you can choose the different modality and structure. Resting state function, MRI, DTI, or task, etc. Once you click a queue for download, and just download. It just takes up, it's taken quite a long time actually to download. We tried that before. And before we move on to the next one, just want to show there's another very useful button here is the open data sets. Once you click here, it's going to like a cover page of the data, the group you can access. So here there's quite lots of useful information. So for example, the HCP data reference manual. I'm going to refer that a couple of times. So it's very easy to get lost around all the data. But with this one, it's like a manual. You can always find out what is this data, what does that mean. Also, they have, if you're interested, you can have the behavior data for their task, and also, if you're interested in the task, you can have the ePrime script for the task. And also, not only the individual participants, they also have some group analysis that's ready for you to grab. Okay. So the next thing we're going to quickly go through is the connector in the box. So as you can see, this is a few hard drive that we have in our labs back into 2014. This is a 4 terabytes of drive. This contain that the S500 versions of the data. So if you don't want to wait for weeks to download the data, you can order that. It's shipped to you. I still remember the price is okay. You just like order the drive. But the problem is once you have the drive, you have to figure out how to have a computers and plug that in and copy to a local clusters or your computer to process it and figure out how to storage them. So this, I want to mention that because that will set up a big contrast for us from 2014 and 2015 that we know that HCP is mounted on Massive. At that time, Massive now is a CVL M3 from S500, S900, and S12. So all of this data, and then it's help us a lot to facilitate all of the research. Okay, so let's quickly go into here. So what it looks like in how do we access to HCP data on Massive? So firstly, because we need to log on to the Monash HCP ID. So for all of the people who have access to Massive, you will be able to log into this system and then go to the software agreement and then add software. And then once you click on that, you got a group of software. So HCP data is part of it. As you can see, there's HCP lifespans and etc. So once you click that, it will give you the details of how do you access, how do you get access. So simply for that, you just need to register it and then using your email address that you provide to link to with your Massive account. And then once you get the granted access to HCP data, you will be automatically get the access to the HCP data on Massive. All right, so this is how this looks like. It's quite straightforward. So all of the data is hosted on M3 Scratch. And then here's the drive. Just on the FS03/ACP1200. And once you open here, this is each individual's folders, and then it's all in there. And then there's read writes, get a read writes for all of the HTTP data, all of the group members within the HTTP data group. Okay, because unfortunately Massive is on a regular service today, so I just screen record a lot of things, and then I can just go through with the videos. So here is basically showing the location and take a browse of what is HTTP data looks like. Remember, the data is in FS03 HTTP12, and we just open the random persons, and we see the next level folder. So we have preprocessed, and preprocessed. So for example, that's the raw data. If we put in the list view, we can see all of data in the zip format. So diffusions, resting state, task, different tasks, and then come to the 70. So some people have a different modalities than the others. So they have a 70 data. Basically, everything is in there in the zip format. And then the pre-processing is the same things. You can take a look at it. So we have everything's in there. We put it in the zip format just to save space, but I think when next year, when we upgrade the storage system, so we will put them into unzipped format, so it make it even easier for researchers to access. And for this particular person, they have MEGs and the fixed standards, the resting state data. As I said, different people coming from a different release, so they will look slightly different. For example, this person doesn't have the MEG data, and it doesn't have 70 data. So that's why when you get access, you probably should know what the modality you want. You get the proper list of the participants you want, and then extract the data yourself carefully. Right. And then I would say there are two important documentations in here. The reference manual, maybe Catherine can help put into the chats later before. So these will help you just to, once you get confused within the data, each folders, then this is a good manuals to help you. Okay. So as I said, everything is zipped. So the first step you're going to do, and we're going to do it during the hands-on session, is to unzip the data. And then take examples of these folders. After unzipped the preprocessed folders, we actually have the MNI space, T1 space, and some release note here. So next we're going to just quickly touch base into how do we view this data. So once we unzip the data, they just easily to be accessed. So let's just start with, if we just want to have using the basic FSLIs to view the data, you're going to, for example, we want to use the T1 weighted data, and then that's in T1 is in native space. And then we open a terminal. Remember, all of the software has been preloaded on the Massive. You just need the module load FSL and open FSLIs, and all of the other software that you can think that you probably have your favorite software to view the data, like FreeView from FreeSurfer or the viewer of MRTrix, etc. They're all preloaded. Okay, so then you just need to browse the data, and then, for example, you want to looking at the T1 data, so they just loaded there. Let's just quickly skip through. If you open, yeah, so the T2 can be loaded, and then you can play with the transparent bit of the first layers, and you can see there's two files is lined up. And there's some other folders, for example, this native. Once you double click it, you see some files end up with .gii. This is the surface-based image or the vertex-based. So this is projecting the surface. So once you load it, it's not loaded in this view straight away, because this is a voxel-based, which is the volume. We're talking about the volume here. So to view that, an FSL, you go view and then 3D views. In that case, you will see the surface will be rendering in this view, and you can rotate around. And also the T1 and the T2 is also rendering in this 3D space. Because the one they are loaded in the video here is inflated native space, so it's not really matching quite well. So we can remove it, and then we go to that native folders again. Yeah, and we find there's a pause native surface, the left side, and open it. Give it 2 seconds to update. Yep, again, it's not on the volume, but on the surface space, you will see the surface is loaded around, and you put the T1 on, you'll see they match up perfectly. Okay, so all of this I'll share the slide, and then all of this we can actually play with it when we have the on the hands-on sessions. So the next one will quickly show the examples of the resting state. This is the function MRI data. So say we're loading a structure image here, and we go to the results, and this is all of the tasks that has been pre-processed, and we can select one of the motor task, and then just click open, and you will see there's another layers load up, which is the function MRI data, and you will see it's lined up properly. You can make it a little bit transparent, and you can see the data is all in MNI space. That's all pre-processed, ready for the another analysis. Yep. So you can see that's it's going. There's 40 data. That looks pretty good. Right. Quickly, next one we're going to talk about is the Connectome workbench. So it's coming with the Connectome Human Connectome projects, and then the working project is a visualization platform using C+ software developed. So there's a big emphasis in the HCP data about the surface and then vertex-based analysis. So this software is really good of combining the volume-based and the vertex-based visualization together. It's a quite complicated software, but if you get lost, again, they have a really nice tutorial, and there's annotations and used to show you all of the buttons. How does this work? Okay, so let's just quickly go into an examples of viewing a structured data. So for example, after unzipped my pre-processed data, I go to MNI space. This folders, we have so many data in here that if it's the first time you're using it, you will get lost. But I will draw your attention to all of this .spc files. It's actually like a little environment. Once you load that, it helps you to load all of the necessary the files that you want to load it. So they have another similar .spc files in the native space as well. I'll just skip through that. It's just showing where's the file it is. And then that's start the HCP workbench, which is in just the pop-down menus in here. Again, all of the software has been preloaded, pre-installed on Massive CVL. CVL at Massive, so it's quite easy. So you can see we have a lot of things preloaded, but this time we're just showing with the open a new one. So we navigated to the folders. Yeah, and then we can select the pre-processing in MNI space. So there are two spec file. It's just a different way of color registration or normalization. So let's choose the easy one. Let's go to the native space. Oops. Yeah. So once you click and load the file, so you will see all of the files will be loaded for you. So that's all of the file will be loaded. So once you load that as .spec files, all of the file will be loaded together, and it will bring you to this complex interface. But before that, I will just draw your attentions. It is complex, but there were 5 important tabs in here. So which is the voltage and the volume base, etc. So in the volume base, you have all of these. Oops. Yep. So in the voltage base is actually just the surface base. So you have a different layers that you can put on. So you can have, for example, you want to put this layer out with the myelination map. Basically, that's a T1, T2 ratio related measurement. You can move that layer up or that down. And also you can change the transparent, very similar like FSL eyes. And also you can play with the distribution, the threshold, and the histogram, and then the color scheme. So all of that, you can do it. And then once you're happy with it, this is the image that you can view. And also you can load the different layers of, say, thickness, verification information, etc. think just a matter of the time. So we're running out of time, but I'll just quickly browse through the rest of few slides. I'm going to show you the task-based function MRI. That's what we're going to work through, and then to show you how to load that in the workbench, and also the resting state. This is basically the melodic ICA output that you just grab it after you unzip the fixed extent folders, and then how to get the beautiful visualizations. Let's quickly go through, and also we can also checking the DTI data after they're running the Batpost X. Sorry, because of the time, I couldn't show everything to you, but please joining in 15th of December. We're going to spend a little bit longer to go through that. Also, if you have research ideas, you can feel free to bring it and then browse the HCP data and then start get a feel of how easily you can get access. And then please stay tuned of the HCP series of workshop in next year. Just check this website or email Catherine or myself to get more information.
Thank you very much, Chao, for this great overview. So yeah, that would be great. I think many people are looking forward to get their hands on this data and play with it. So yeah, with this, we're perfectly in time to finish this webinar. So I thank again every speaker that presented today. It was really cool to get this great overview, and the organizers and getting everything ready for this cool session. So thank you very much, and see you next time.